Today, we are going to learn about the proteomics and metabolomics. So, firstly, we will start with proteomics, and then we will see what's metabolomics called and how they are like helping us. Okay, so let's start. So today we are going it's the table of content. So what we are going to study today: proteomics, techniques in proteomics, its application, tools, then metabolomics, then integration of both of them, then what's multi-omics and system biology approach, and case study and future direction, and then MCQs. So let's start with proteomics. Do you guys know what is protein? What's the building block of proteins? Let's start with the basic. What do you mean by proteins? Then I will tell you what is proteomics. Yeah. So see, I have talked about central dogma many times, like in our previous lectures. DNA replication, then it transcribed to RNA after transcription, then it gets converted to proteins, right? So proteins are made up of amino acids, right? So how many proteins are present in our body? The thing is, it's an ongoing research topic. It's not like exact number is still unknown, but 20 to 25,000 proteins are present in our body. And if we talk about like the post-modification, like post-transitional modification, there are around 1 million proteins. Okay. Any doubt? We have not started yet about the proteomics. I am talking about proteins. So what are the different functions performed by proteins? See, proteins are our building blocks, which helps in repairing of cells. Then enzymes are made of proteins, right? Most of the enzymes are proteinaceous in nature, right? Then in our immune responses, proteins are helpful. Antibodies present in our body are proteins, right? Then maintaining the fluid balance, then energy and transport, proteins are used. So how many amino acids are there? If we talk in general, how much you guys know about proteins? Yeah, there are 20 amino acids. Can you name any of them? What are the 20 amino acids? These are like alanine, arginine, asparagine, aspartic acid, cysteine, glutamic acid, right? Leucine, isoleucine, methionine, right? Have you guys heard these names earlier? These are the 20 amino acids. As you are from biology background, I think you are aware of these things, right? Okay, so there are 20 amino acids, and these 20 amino acids are formed by 64 codons, right? And then there are millions of sequences of these proteins, like their arrangement. Okay, so what are the different level of protein structure? I have like put a diagram of different amino acid structure, like at the primary level, secondary, tertiary, and quaternary levels. These are the 4 levels of protein structure, right? So if we talk about, now we are switching to proteomics, right? So have you guys ever heard this term proteome, and what does it mean? See, study of proteome is proteomics. Now you have to tell me what is proteome. See, protein plus genome forms proteome. All the proteins present in a cell or in tissue or in any organism is proteome. Okay. Any doubt? Any doubt? Proteome means protein plus genome. All the proteins present in a cell, proteome. Okay? See, now let's switch to this. Proteomics means study of proteome. Proteomics is a large-scale study of proteins focusing on their structure and function. If we talk about functions, there are different function for different protein, right? If we talk about insulin, what's the function of insulin? We'll talk about insulin today. Insulin is a protein, right? Have you guys heard about insulin, glucagon? These are hormones. So proteins act as hormones as well, right? Then the goal to understand, it controls the sugar level, blood sugar level. So let's see, the goal to understand the protein content of cells, tissues or organisms and their roles in biological processes, including disease, metabolism. We are going to study about these things today. What are the key goal of proteomics? Why we are studying this? Understanding disease mechanism. So proteomics plays a key role in understanding the molecular basis of disease such as cancer, Alzheimer and diabetes. So we can identify the altered sequence of the protein expression or any modification, if any modification causes any kind of disease. Okay. So for disease, now take an example. So I was talking about insulin. The function of insulin is controlling the blood sugar level, right? So insulin is present in our body. If it's not working properly, then it will lead to diabetes, right or wrong? If insulin is not working properly, then it will lead to diabetes. Okay. So now comes the role of proteomics. We will identify why the insulin protein is not working. It is made correctively or not. Means the structure of insulin present in a diseased person, we compare it to the healthy person, right? Okay. Firstly, we identify the insulin protein, like it's made in correct order or not. Then what we will do is analyze it, how insulin interacts with other proteins in the body. Then we will detect if there is any changes in the structure and function. Then it might cause problems, right? Then what can we do now? Then we can develop new treatments and create personalized medicine, right? So first goal is understanding disease mechanism. We can identify, analyze and detect. The second thing is drug targets and development. We can identify, see, whenever we consume any medicines are mostly drugs, right or wrong? Medicines are drugs, right? Amoxicillin and all, azithromycin, paracetamol. These are different types of drugs and antibodies, antibiotics we are using in our day-to-day life, right? So what are drug targets? Whenever we consume any drug, then it binds to a specific molecule in our body, right? How the drug is interacting in our system. So identifying potential drug targets by exploring proteins that contribute to disease processes. Protein implicated in diseases are often the focus of therapeutic intervention. Okay, we can make medicines only when we know where how will it react in our body, right? Any doubt? Are you guys able to understand whatever I'm saying? I have talked about proteins, then proteomics, then what are the key goals of proteomics. Then first point is understanding disease mechanism. The second thing is drug targets and development. Okay. Okay, good. So we identify new target for drugs in our body. Then we understand how proteins interact with each other and with the drugs. What are the different interactions we will do in detail. Then biomarker discovery. We talked in genomics and transcriptomics about biomarkers. What are biomarkers? What are biomarkers? Biomarkers are like biological fingerprints, which help us to diagnose or monitor diseases. In case of genomics, whenever we do DNA profiling or DNA fingerprinting, there are some genetic markers. What are those? What are genetic markers? STRs, SNPs, right? Understood? So we identifying protein biomarkers that can be used for early disease detection, prognosis, or monitoring treatment responses. Okay? Any doubt in any point? I'm touching the topic from the basics, from the protein, from the different structures of protein, what are the different functions in our body. Biomarker, okay. See, you guys heard about genetic markers, right? So, biomarkers are like biological fingerprints that help us to, these are the different sequences, I can say. In case of genetic markers, what the different biomarkers were? SNPs, STRs, YSTR. So these are different. Okay. So these help us in detecting diseases. We can monitor treatment responses. Okay. These are like specific molecules present in like our body. You will understand this term in like after a few slides where I touched the topic of cancer proteomics. Okay. Now we can move to the next slide, right? Okay. Move to the next slide then. So these are the protein structures. If there is a chain of amino acid, the structure is known as primary structure. Then if alpha helix, beta helix, these are the secondary structure. Then there's a polypeptide chain and it's a 3D structure, tertiary structure. Then aggregation of these two or more polypeptide, quaternary structure. Right? These are the different levels of protein structure. Right? All clear? Okay. Then proteomics workflow. So during genomics, what we did was sample preparation was there. We extract the DNA from the whole sample, right? We extracted the DNA from the cell after cell lysis. So in the same way, I think my slide is on what is protein. Okay, let me change the slide first. So I was showing this and my slide was not changing. Okay, now. So these are the different protein structure. And building block of proteins are? What is the building block of protein? Write in the chat box. What's the building block of protein? Write in the chat box. You guys should be interactive with me. Yeah, amino acids are the building block of proteins. If it's a simple chain of amino acid, primary structure, and if it's alpha in the form of sheets, alpha helix and beta sheets, then secondary structure. Then the overall 3D shape of the protein resulting from the interaction between amino acid and the secondary structure element, then tertiary structure, then arrangement of multiple polypeptide chains. It will form quaternary structure, right? Okay. Then next slide. Proteomics workflow. So when we did genomics, what we did was about the DNA extraction. After cell lysis, we extract the DNA, right? In the same way, firstly you will prepare sample for the proteomics. So first step is sample preparation. The first step involves the isolation of protein from the biological sample. See, in a cell, DNA is present, RNA is present, proteins are present, and different cell organelles are present. So our task is to separate the protein. We have to isolate the protein from the cell. So the proteins are typically extracted, purified, and sometimes denatured to simplify further analysis. Okay. So what do you understand by denaturation of proteins? What is protein denaturation? Nobody know what is protein denaturation? When we unfold the protein structure, we talked about primary, secondary, tertiary, quaternary structure. When we unfold the protein structure now, so how it can be done? We can by giving high temperature, right? By high pH, we can use chemical denaturants. Then we can provide mechanical stress, right? The unfolding of protein molecule is known as denaturation, right? It can be done using high temperature, pH and all. Yeah, we are changing the conformations. Okay. So, common techniques in sample preparation is homogenation. We do cell lysis. Precipitation is done, same as DNA and RNA we did in our previous class. Then protein quantification. So first thing is sample preparation. We have to isolate the protein. Second thing is protein separation. Proteins are separated based on their sizes, charge, or other chemical properties, right? We as we were doing electrophoresis in case of DNA. Okay, then mass spectrometry. The separated proteins are analyzed using mass spectrometry. So, in mass spectrometry, the molecules are separated on the basis of their mass by charge ratio, right? Then, the mass to charge ratio of the ions provide information on the protein's identity and structure. Then, what are the key steps? Ionization, fragmentation, and detection, right? Then data analysis. The resulting data from MS are processed and analyzed, and using bioinformatic tools, we can read the sequences and all, right? There are different bioinformatic tools. So have you guys opened those tools in your laptop or not? Today I will give you a homework. You have to download some tools related to the proteins and metabolites, like metabolomics we are going to study now. So what you have to do is you have to download those tools, and then you can pick any sequences from the ChatGPT or other platform, and then you can play with those tools. Only then it will be beneficial for you. Okay. So these are the lab techniques which we basically use. We are using MS, NMR, right? Okay. Any doubt? Any doubt? No doubt. We can move to the next slide, right? So, in like easy terms, proteomics workflow is firstly sample preparation, then protein separation. Then we can use any technique to separate them, like MS and all. Then we can analyze the data. Okay. Then techniques in proteomics. These are important. So all of these are like lab techniques. We are going to study about 2D gel electrophoresis, then chromatography, then mass spectrometry. Okay. See, what's the purpose of these techniques? Why we are using these techniques? Why we are using these different techniques? To separate protein based on their different factors, right? So 2D gel electrophoresis. If we talk about gel electrophoresis, it means you need gel to separate molecules. Basically, two gels are used. One is agarose gel and the other one is polyacrylamide gel, right? You guys heard these names earlier, right? Agarose gel electrophoresis. Agarose gel electrophoresis is used for, where you heard this term? आप लोगों ने सुना होगा ना agarose gel electrophoresis. सुना है DNA के केस में? For DNA, RNA, we use agarose gel electrophoresis, and for proteins or small size DNA, what we will use polyacrylamide gel. Polyacrylamide gel electrophoresis. Basically, see, gel electrophoresis, gel plus electrophoresis. We are using a gel, and electrophoresis means electric current is used. Okay, so it's a technique that separates proteins based on two factors. What are these two factors? Isoelectric point and their molecular weight. The pH at which a protein has no net charge, first dimension, and then on the basis of the size of protein, second dimension. So what are the application of gel electrophoresis? It allows the separation of complex protein mixtures, helping to identify and compare protein expression levels under different conditions. Okay. What are the limitations of gel electrophoresis? Limited in its ability to separate large proteins and is not effective for membrane-bound or hydrophobic proteins. So, it's in short, right? Then, we move to the next technique, chromatography. See, you guys have to do these like techniques in detail. Basically, it's a 2-hour lecture. I am not able to cover each and everything in detail, but I try my best so that you guys can understand it in an easy way, right? So chromatography. Then this technique is used to separate proteins based on their physical or chemical properties, right? We are using these techniques like for specific protein isolation. We purify on the basis of their size. See, on the basis of size, which chromatography technique will be used? Size exclusion chromatography. And on the basis of ions, we can use there are different types of chromatography, right? Have you guys heard about the different types of chromatography? Write any name in the chat box, like about the techniques, chromatography technique. We can separate on the basis of chemical properties, on the basis of size, charge, hydrophobicity, affinity. Yeah, on the basis of affinity chromatography, right? Paper chromatography, thin layer chromatography, column chromatography, right? So you guys have to cover these topics in detail. I don't know about your syllabus. And if there is any need, you guys can do. Then types of chromatography used in proteomics. If we talk about proteins, we can use size exclusion chromatography. Separate on the basis of size. Ion exchange chromatography on the basis of their charge, right? The charge on the ions. Then affinity chromatography based on the specific interaction with the ligands, right? Where we can use this used in conjunction with other techniques like mass spectrometry to purify proteins and for detailed analysis. Okay. We have covered chromatography and what was the previous topic of our slide? I'm watching. You guys are active or not? What was the topic of my previous slide? Now we are reading chromatography. Proteomics, right. But I was talking about one more technique now. Gel electrophoresis. 2D gel electrophoresis. Okay. Now, I was talking about this slide, 2D gel electrophoresis. What are the different techniques in proteomics? 2D gel electrophoresis, then chromatography, then we will talk about mass spectrometry. So, what is mass spectrometry? It is a key technique in proteomics used for protein identification and quantification. It measures the mass to charge ratio of ions generated from the sample. So, what are the different types of mass spectrometry? See, it's the full form. MIDTO. There's a question in your MCQ. So, remember it. It's for matrix assisted laser desorption ionization, time of flight. So proteins are ionized using a laser in the matrix and the ions are analyzed based on their time of flight. Okay. So remember this. It's a type of mass spectrometry where we are using laser in the matrix and the ions are analyzed based on their time of flight. Okay. So its application is used for the protein identification and rapid analysis. Then ESI, electrospray ionization. Proteins are sprayed into fine mist of charged droplets. The ions are analyzed based on their mass to charge ratio. So basically the principle will be the same, mass to charge ratio. Then application, better for analyzing large biomolecules and peptides, often used in quantitative proteomics. Okay. Any doubt? So we talked about three techniques. One is gel electrophoresis, second one is chromatography, third one is mass spectrometry. So for proteins, we use matrix assisted laser desorption ionization, time of flight, and electrospray ionization. Okay. Now move to the next slide. So this diagram is of which technique? You guys have to tell me. The diagram is of which technique? You are taking a protein mixture and then isoelectric focusing, then pH is changed, separating first dimension by charge, then in second dimension by like pH or size, then apply first gel to top of second. This technique is called, what's the name of this technique? Tell me in the comment box. You guys are not able to identify it. It's clearly mentioned here. STS, electrophoresis. So this diagram is of STS electrophoresis. Right? Understood? Yes or no? Okay, you guys are not getting my points, I guess. Understood? Yes. Okay. Then it's a protein affinity chromatography. So it's a mixture of protein. Then these are the target proteins. Then ligands is there. Other proteins are there. then affinity resin with ligand attached. Like you can purify your sample of proteins, right? Then protein-protein interaction studies. So protein-protein interactions are critical for understanding cellular processes, right? Including signaling pathways, metabolic networks, and disease mechanism. So we have to identify the interaction between proteins, right? We are using two key techniques. One is yeast-2 hybrid, then Co-immunoprecipitation. So we are going to study these two techniques in detail to study protein-protein interaction. Firstly, I want to show you the diagrams. So in this technique, just focus on the principle. It detects physical interaction between two proteins using yeast cells. We will use two types of binding domains. Processes split the transcription factor into two domains. One is DNA binding domain and second one is activation domain. Then fuse one protein of interest back to binding domain and another to prey to activation domain. Now if the two proteins interact, the transcription factor reconstitute and activates a reporter gene. Okay, see, simply हम इसको देखते हैं. हमने दो वो लिए. One is DNA binding domain and other one is activation domain. Okay, अब हम क्या करेंगे? I think I can use Hindi here, right? For your easy understanding. प्रोमोटर पे हमने DNA बाइंडिंग डोमेन को अटैच करा. यहाँ पे हमने एक्टिवेशन डोमेन है हमारे पास. अगर X और Y दो प्रोटीन्स है, यहाँ पे X and Y are two proteins. अगर इनकी इंटरेक्शन हुई तो आगे ट्रांसक्रिप्शन होगी. ट्रांसक्रिप्शन will take place if these two proteins interact with each other. Okay, then इंटरेक्शन हुई तो ट्रांसक्रिप्शन हो जाएगी. And if transcription took place, we can attach RNA polymerase 1, RNA polymerase 2. Okay, you guys have to tell me what's the function of RNA polymerase enzyme 2? RNA polymerase 2 enzyme function. What's the function of this enzyme? You don't know the answer? Yeah, obviously it's helping in transcription. It transcribes DNA into RNA. Right? Right or wrong? And what's the other function? What's the other function? I think there's an enzyme which reads the template. Okay. So basically, what is to hybrid system technique? We are seeing the protein interaction here. यहाँ पे हम देखेंगे अगर प्रोटीन की इंटरेक्शन हो रही है तो ट्रांसक्रिप्शन आगे होगी. और सी यहाँ पे X और Y दो प्रोटीन्स हैं. ओके, BBD binds to DNA in promoter region and protein Y interacts with protein X यहाँ पे. और फिर रिक्रूटमेंट ऑफ RNA polymerase 2. अंडरस्टूड? इस टेक्निक में क्या होता है? अंडरस्टूड और नॉट? One is bait and one is prey. अगर प्रोटीन इंटरेक्शन होती है तो आगे ट्रांसक्रिप्शन होगी. नहीं समझ में आया? Saharish Gupta, you are not able to understand what I am saying. हाँ हमने प्रोटीन रीजन पे, प्रमोटर रीजन पे हमने व्हाट क्या बाइंड करा है, डीबीडी, डीएनए बाइंडिंग डोमेन। दूसरे रीजन पे क्या है, एक्टिवेशन डोमेन। हमने प्रोटीन डाला X और Y। अगर इन दोनों की इंटरेक्शन होती है तो आगे ट्रांसक्रिप्शन होगी और आरएनए पॉलीमरेज 2 यहाँ पे रिक्रूटमेंट हो जाएगी। और यहाँ पे अगर नहीं होती, एक दूसरे से अगर बाइंड नहीं करेंगे, नेक्स्ट एग्जांपल इज ऑफ दे आर नॉट इंटरेक्टिंग, देन प्रोटीन Y does not interact with प्रोटीन X, so no recruitment of RNA polymerase। यहाँ पे बाइंड ही नहीं करेगा तो आगे ट्रांसक्रिप्शन नहीं होगी। ओके, so let's start it from the start. So IST2 hybrid system. So the principle of this technique is it detects the physical interaction between two proteins using IST cells. Okay, the process is split a transcription factor into two domains. हमने दो domains में divide कर लिया. One is DNA binding domain. The second one is activation domain. Okay, fuse one protein of interest to BD, मतलब बाइंडिंग डोमेन पे हमने एक प्रोटीन डाला, प्रोटीन ऑफ इंटरेस्ट, और दूसरा हमने डाल दिया किस पे, एक्टिवेशन डोमेन पे, एडी पे, तो उसको हमने बोला प्रे। इफ द टू प्रोटीन्स इंटरेक्ट, द ट्रांसक्रिप्शन फैक्टर रिकॉन्स्टिट्यूट्स एंड एक्टिवेट्स अ रिपोर्टर जीन। अब हमारा क्या हो जाएगा, रिपोर्टर जीन एक्टिवेट हो जाएगा अगर इन दोनों का आपस में इंटरेक्शन हुआ तो। आया समझ में? अंडरस्टूड? एनी डाउट? अभी समझ में आया नहीं आया? See, now move to the applications. Mapping interaction network. Then it helps in mapping interaction networks. Then identifying unknown partners for proteins. Then advantages, high throughput and cost effective. Then what are the limitations? It can give false positive result as well. Limited to interactions that occurs in IST. Okay. Then one more technique is there where we can see the protein-protein interaction, and it's known as Co-immunoprecipitation. So what's the principle of this technique? See, the principle is it captures protein complexes by using antibodies specific to one protein. So what we have to do is, the process is lyse cells and incubate lysates with an antibody targeting to the protein of interest. हमें सेल को मतलब lysis करना है, then we will add an antibody here, then centrifugation. See, use bead coated with protein to pull down the complex. Then analyze the associated protein using SDS page or mass spectrometry. Application, validating PPS. Firstly, see the diagram. What is there? Protein complex is there. Then protein primary antibodies, secondary antibodies of protein, agarose beads and magnetic beads are there. Okay, then washing, elution, and you can read if interaction between protein and antibody took place, then you can detect it on STS page or Western blotting technique, right? यहाँ पे हम क्या देखते हैं, प्रोटीन और एंटीबॉडीज के बीच में अगर इंटरेक्शन हुआ है या नहीं हुआ है। हमारे टारगेट प्रोटीन है एंड एंटीबॉडी स्पेसिफिक एंटीबॉडी है। ओके, अंडरस्टूड? ये टेक्निक समझ में आई, को इम्यूनोप्रेसिपिटेशन टेक्निक। in vivo technique, understood or not? Agarose shell to pull down the complex. Okay, at least send a thumbs up. समझ में आई तो we can move to the next. You are saying समझ में नहीं आई, ऐसे ही? खुश अरोड़ा, what are you saying? See, यहाँ पे नॉर्मल हमारा क्या प्रिंसिपल है, हम एक प्रोटीन लेंगे, टारगेट प्रोटीन, और एंटीबॉडीज लेंगे, हम एंटीबॉडीज डालेंगे राइट सैंपल में। अगर वो एंटीबॉडीज प्रोटीन के साथ रिएक्ट कर गई तो हमें पता लग गया ना कि इंटरेक्शन हुई है। अगर वो प्रोटीन एंटीबॉडी मतलब एंटीबॉडीज प्रोटीन के साथ बाइंड नहीं करेंगी तो मीन्स इंटरेक्शन नहीं हुआ उन दोनों का। और इस चीज को हम कैसे देख सकते हैं, मतलब मास स्पेक्ट्रोमेट्री से और एसटीएस पेज टेक्निक से राइट, इंटरेक्शन हुई है या नहीं हुई है। सो को-इम्यूनोप्रेसिपिटेशन टेक्निक। ओके या फर्स्टली सेल लाइसिस करेंगे, फिर एंटीबॉडीज को ऐड करेंगे, बट इसमें क्या है, हमने यूज कर रखा ना मैग्नेटिक बीड्स लेंगे, हम बीड को मतलब मैग्नेटिक बीड्स लेंगे हम। ओके इनका इंटरेक्शन वी आर लाइक गोइंग टू सी। Then what are the applications? Validating PPI is identified by high throughput methods like Y2H. इनका throughput ज्यादा होता है, means studying protein complexities in their native states, right? Limitation require high quality antibodies and it's time consuming. Okay. Now move to the next topic, functional proteomics. Here we are focusing on the studying the biological roles and activities of proteins, including enzymatic activities and cellular signaling pathway. What are the key areas? Decoding enzyme activity. So there are different enzymes which are proteinaceous in nature, right? So protein, particularly enzymes, drive many biochemical reactions. Functional proteomics investigate their activity under different conditions. So as we know, जो हमारे एंजाइम्स होते हैं, they work at a optimum temperature and optimum pH, right or wrong? एंजाइम्स optimum temperature और optimum pH पे काम करते हैं, जो उनका एक फिक्स होता है, right? Right or wrong? So functional proteomics में हम क्या करेंगे, हम उस चीज पे फोकस करेंगे, उनके जो बायोलॉजिकल रोल्स हैं, लाइक एंजाइमेटिक एक्टिविटीज है। हमें पता है एंजाइम्स, मोस्ट ऑफ द एंजाइम्स वो होते हैं प्रोटीनेशियस होते हैं, प्रोटीन के बने हुए होते हैं। तो उनके फंक्शन के बेसिस पे जब हम उसको फोकस कर रहे हैं तो फंक्शनल प्रोटियोमिक्स। सो की एरियाज डिकोडिंग एंजाइम एक्टिविटी, टेक्निक्स सच एज एक्टिविटी बेस्ड प्रोटीन प्रोफाइलिंग वी कैन डू। These are used to study enzyme function and regulation. Okay, these studies help identify enzymes that are potential drug targets, particularly in metabolic disorder and cancers. Enzymes के cases में you guys must heard about the loc and key hypothesis, जिस चीज पे enzyme काम करते हैं, right? So functional proteomics में हम enzyme की activities पढ़ते हैं and signaling pathways. Proteins involved in signaling pathways are often studied to understand cell communication and response to external stimuli. Functional proteomics identify proteins involved in phosphorylation, ubiquitinations, or other modification that regulate signaling networks. For instance, proteomic studies have revealed key insight into a MAPK ERK pathway, which is crucial in cancer progression. Right? Okay. So, cell communication. Have you guys heard about like stimuli? What is stimuli? Stimuli क्या होती है? Have you guys heard about the touch me not plant? Whenever we touch that plant, it like closes its leaves or something, right? See, जो भी like environmental conditions होती हैं, उसमें हमारा like response क्या होता है सेल का, to understand that thing. Stimuli से ही हमारा response आता है, right? So you guys knows about phosphorylation. What is phosphorylation? Addition of which group? And there is one more term written here, ubiquitination. So what's the meaning of this term? Yeah, addition of phosphate is known as phosphorylation. And there's one more term here. जो पोस्ट ट्रांसलेशन मॉडिफिकेशन होते हैं जहाँ पे हम यूबिक्यूटिन प्रोटीन हम ऐड कर देंगे, so उसको हम यूबिक्यूटिनेशन बोलते हैं. Okay. Then signaling, for instance, proteomic study have revealed key insight into the MAP के pathway and ER के pathway. These are like these follow different pathways, signaling pathways. आपने कभी वो पढ़ा है जो हार्मोन्स के कब कौन सा हार्मोन एक्टिव हो रहा है, तो इनमें सिग्नल्स आते हैं ना. In case of hormones, signals are sent in our body. So on the basis of this, they are like inhibition is there. कब किस हार्मोन का सिक्रीशन करना है, तो signals are sent ना. Okay. So there are like many pathways are there. The signals can be from outer environment or from the inside of the cell. Switch on and switch off processes, right? Yeah. So signals are sent. कौन सा हार्मोन कब like इसी वे में जो प्रोटीन्स के जो सिग्नल्स होते हैं, तो signaling pathways in the case of proteins. So signal can be from outside, right? Signal can be from if any like receptor bind to any site, so it can also give the signal. Okay. Then we can move to the next slide now. Clinical proteomics. So, clinical proteomics applies proteomic technique to understand disease and identify biomarkers. Biomarkers are molecules that indicate the presence or severity of a disease. Okay. So, cancer biomarkers. Proteomics has been instrumental in discovering biomarkers such as PSA, prostate-specific antigen for prostate cancer, and then techniques. We can use different techniques like MS and immunoassays, which are used to identify proteins that are like responsible for these. I think this line is incomplete here in my slide. Differentially expressed in healthy versus diseased tissues, right? So clinically significance क्या होती है कि हम इसको मेडिसिनल फील्ड में हाउ कैन वी यूज दिस टेक्निक, यूज दिस लाइक structures. So cancer biomarkers. So we can study about the sequences of proteins and different like genome, right? What will we do is we will like match it with the healthy person. हम healthy person और disease person में check करेंगे ना कि क्या difference है इनकी उसमें. Then we can identify. So किस चीज की बेसिस पे हम like वो करते हैं identify biomarkers की हेल्प से. ये molecules होते हैं, इनकी जो presence है या severity है. So in Alzheimer patients क्या होता है, tau protein होता है एक, तो that is more in the cases of the diseased person. Okay, so but we can analyze from it. अगर कोई प्रोटीन ज्यादा है और वो कोई like डिजीज का सिम्टम दे रहा है हमें, डिजीज पर्सन में अगर कोई प्रोटीन ज्यादा है तो इसका मतलब वही है ना, हमारे क्या होता है कोई चीज ज्यादा भी like अच्छी नहीं रहती और कम भी अच्छी नहीं रहती, राइट? तो वो डिजीज कोज करते हैं. अगर वो उसका लेवल ज्यादा है तब भी डिजीज है, उसका कम है तब भी डिजीज है, तो मॉडरेट अमाउंट में होना चाहिए, राइट? सो कैंसर बायोमार्कर्स आर देयर. We can use proteomics in this field as well. Then neurological disorders. In diseases like Alzheimer's, clinical proteomics identify protein aggregation or altered protein levels in cerebrospinal fluid. I was talking about this. Any doubt? Any doubt? Okay. At least write in the chat box. If you want me to repeat any point, then I can. Okay. Okay, then move to the next slide. Then these studies have highlighted proteins like tau and amyloid beta as key biomarkers. So biomarkers वही हो जाएंगे कोई specific molecule if its concentration is high in any cell or if its concentration is low, then we can like analyze it. Personalized medicine. Proteomic data are increasingly used to tailor treatments based on individual patient profile. Then, for example, HER2 protein expression in breast cancer guides the use of targeted therapies like trastuzumab. So basically, what it is personalized medicine. से आप क्या समझते हैं? What do you understand by personalized medicine? We use these terms many times, right? Right or wrong? Personalized medicine helps, like it's a key application in the field of bioinformatics. Nobody is responding. Why? See, we talked about cancer as well. Yes. अब वो इंडिविजुअल पर्सन के लिए मेडिसिन क्यों बनाते हैं? ऑब्वियसली कुछ अगर हम उसका सीक्वेंसेस को उसके डीएनए या प्रोटीन्स या आरएनए हम किसी भी उसको उठाते हैं लाइक मॉलिक्यूल को, इफ इट्स लाइक डिफरेंट फ्रॉम द लाइक हेल्दी पर्सन, तो हेल्दी पर्सन डिजीज पर्सन में क्या डिफरेंस है? कहाँ पे डिफरेंस आया? कहाँ कौन से सीक्वेंसेस में हम वो करते हैं? तो उस जो हम उसके लिए अलग से वो कर रहे हैं, तो दैट इज पर्सनलाइज्ड मेडिसिन. सो HER2 प्रोटीन, we talk about cancer is uncontrolled growth of cell, right? So we talked about HER2 protein. So क्या देखा गया? जो कैंसर सेल्स हैं उनमें ये वाला प्रोटीन है जो HER2 वो ओवर प्रोड्यूस हो रहा है, मतलब ज्यादा अमाउंट में. द कंसंट्रेशन ऑफ HER2 प्रोटीन इज मोर. सो हाउ कैन वी यूज प्रोटियोमिक्स हियर? We can study the structure and function of HER2 protein here. We can identify the regions of protein that can be targeted by drugs. So how to control the growth of like these protein, right? HER2 protein. Then we can like develop drugs like target therapies like trastuzumab. Okay. Any doubt? Any doubt? देखो बेसिकली बात क्या है, अगर कोई चीज हमारी बॉडी में ज्यादा बन रही है फिर हम क्या करेंगे? हम ड्रग्स डेवलपमेंट, जो बायोइन्फॉर्मेटिक्स की एक और एप्लीकेशन है, टारगेटेड ड्रग, किस प्रोटीन पे और कौन सा ड्रग काम करता है, मतलब कौन सा ड्रग किस प्रोटीन पे काम करता है, हम उसको देखेंगे. अगर कोई चीज ज्यादा सिक्रीट हो रही है, हम बाहर से ड्रग्स डालेंगे बॉडी में, वो उनके साथ इंटरेक्ट करेंगे, उनको न्यूट्रलाइज कर देंगे, तो वो एक क्या हुआ? ट्रीटमेंट हुआ उस चीज का. आप सब हिंदी में कंफर्टेबल हो? अगर कोई पॉइंट में लाइक मैं सोच रही हूँ कि इट विल बी इजियर फॉर यू गाइस टू अंडरस्टैंड इन हिंदी, बता देना कहीं कोई फिर बोले बाद में. ऑल गुड? ओके ओके, नाउ मूव टू द नेक्स्ट स्लाइड. Proteomics and drug discovery. So proteomics plays its important role in discovering by processing, by identifying and validating therapeutic target. हम क्या करेंगे? Target identification by following different like techniques. We identify the drug. The techniques such as quantitative proteomics and affinity-based method are used in this step. So the next step is target validation. After identification, what can we do is proteomics help validate the role of these targets in disease pathways. Tools like RNA interference and CRISPR can be combined with proteomics to confirm target functions. Then, drug mechanism study. We can study about the drugs. Like proteomics investigate how a drug interact with its target and its off-target effect. Interaction, like we talked about protein-protein interaction, protein-drug interaction we can study, right? This helps in optimizing drug design and reducing side effects. Then biomarker discovery for drug response. Clinical see, you guys are aware about these terms now, like biomarker and about like their interaction, right? Then bioinformatics tools for, so this topic is of your interest now. So bioinformatic tools for proteomics. The complexity of proteomic data requires specialized bioinformatic tools to analyze, visualize, and interpret the results. Proteome Discoverer. So the interface of this tool is look like this. So आप लोगों का homework क्या है अब? ये वाले जो tools हैं, free demo is like present for these tools. You guys have to download. So यहाँ पे what you can do, what you guys can do, it's a software for MS data analysis. When it enables peptide and protein identification, quantification, and characterization. So you can use these tools. हमने last जो lecture था उसमें हमने different tools पढ़े थे genomics को लेके. किसी को याद है क्या क्या tool पढ़े थे हमने? Sequencing की हमने बात करी थी, right? क्या क्या tools थे हमने जो last class में पढ़े थे? What were the different tools we studied in the last class? Okay, I think you guys forgot. Yeah, needle for sequencing, right? Right. So now those tools for like DNA, RNA, right? Now we are talking about proteins. So Proteome Discoverer is a tool. It's a software where you can analyze the data of MS. यहाँ पे आपको वो peaks देखने को मिलेंगी, वो different peaks आती हैं. So it enables peptide and protein identification, quantification, and characterization. It's a user-friendly interface is there and advanced algorithm. It supports large-scale proteomic studies, right? So I show you the like interface of this tool. It look like this. You have to download its free demo and then start playing with it. And then MaxQuant, one more tool like for proteins, right? Widely used tool for quantitative proteomics, particularly in a label-free or select experiment. You can use it. It supports high-resolution MS data and integrates with other tools like Perseus for downstream analysis. So it helps in identifying post-translational modifications and protein-protein interaction. So here's the interface of this tool. हमने लास्ट टाइम अलाइनमेंट के लिए टूल पढ़े थे. डीएनए की सीक्वेंस है, आरएनए की सीक्वेंस है, हम उसको अलाइन कर रहे हैं, उनको कंपेयर कर रहे हैं, राइट? सो यहाँ पे क्या होता है, जो हमारी डिफरेंट टेक्निक्स होती है ना, एमएस हो गई, एनएमआर हो गई, उनका जो डेटा होता है, मेटा डेटा होता है इन टूल्स का, राइट? मतलब कोई भी हमने लाइक प्रोटीन लिया, उसके ऊपर हमने काम करा, उसका आइडेंटिफाई एनालाइज करा, तो उसका मेटा डेटा निकल के आता है. सो बायोइन्फॉर्मेटिक्स का काम है क्या? डेटा एनालिसिस का, राइट? द डेटा व्हिच कम्स इन योर हैंड्स आफ्टर परफॉर्मिंग द होल लाइक प्रोजेक्ट. सो नाउ यू हैव टू एनालाइज दैट डेटा. सो फॉर दैट, in case of proteins, you can use MaxQuant and you can use Proteome Discoverer, right? So download these tools and see their whole interface, from where to upload the file and from where you get result, right? ठीक है ना ये homework? See, मेरे पढ़ाने से अकेले से कुछ नहीं होगा. You guys have to do experiments on your own, right? Okay, I think we can move to the next slide if you guys are not having any doubt. Basically, what you guys have to do is write these names on Google. The first website, you can open it, and then you can put and upload the data in these websites, and then you can get your result. Okay, any doubt? Any doubt? So till now, what we have studied? proteins, their structure, right? Different type of structure is there. Then what are the key techniques? Right, gel electrophoresis, chromatography, mass spectrometry. Then we studied about the protein-protein interactions, yeast to its system, right? Then what was the other technique? Co-immunoprecipitation, right? Then where is the use of proteomics to study the function of like proteins in clinical function, right? Any doubt? Any doubt? No doubt, then send a thumbs up in the chat. Or any doubt, then you can ask. Nobody is responding. Why? Okay. Okay, now let's come to introduction to metabolomics. So you guys heard about metabolism, the chemical reaction which took place in our body. That is called as metabolism, right? The process by which cells convert energy and nutrients into the components they need to function, grow, and maintain themselves. The chemical reactions or all the body reactions where the conversion of energy took place, right? And what are metabolites? Metabolomics is the comprehensive study of metabolites. So these are small molecules such as sugars, amino acids and lipids in biological system. So किसी भी reaction में, किसी भी metabolic process में ये बनते हैं या फिर consume होते हैं. A molecule produced or consumed during metabolic processes, these are metabolites like sugar, amino acid and lipids. So what are the importance? It provides insights into cellular processes, the biochemical changes and disease mechanisms. So metabolites are small molecules produced during metabolism. Okay, so what are the examples? Primary metabolites, glucose, amino acids. Secondary metabolites, alkaloids or flavonoids. So you guys have to tell me what's the difference between primary metabolite and secondary metabolite. Write in the chat box. What's the difference between primary metabolite and secondary metabolite? Nobody is responding. Why? Nobody is responding. Why? See, there's two main differences between primary metabolite and secondary metabolite. For the primary function, for the basic cellular functions, primary metabolites are present. And secondary metabolites have some specialized roles. Okay. Then primary metabolites are universally present, means they will present in all organisms. And secondary metabolites, they are unique and they are specific in which species they are present. Okay. Understood? What's the key difference between primary and secondary metabolite? Okay, these are their example: glucose, amino acid, primary metabolite; alkaloids, flavonoids, secondary metabolites. They act as like intermediates, signaling molecules, and regulators in cells, in cellular processes. Right? Okay. Now move to the next. So, what are the importance and goals of metabolics? Metabolomics. It provides functional insight, complements genomics and proteomics by connecting genotype to phenotype. Application same for disease diagnostic, See, from the very first lecture, the applications are same. The application of bioinformatics are like for disease diagnostic, for drug development, nutritional studies, right? So what are the goals of metabolomics? Understanding biochemical changes, identify how metabolic pathways are altered in response to stimuli, example drug and diseases, right? We can study about the disease mechanisms, detect biomarkers for early diagnosis and prognosis, right? Then personalized medicine means same. Then next slide, type of metabolomics, targeted metabolomics and untargeted. Means targeted, you are you have choose the specific group of metabolites, your focus is on that specific groups only. Then untargeted metabolomics, you don't know about the metabolites, so it aims to profile all detectable metabolites in a sample. Okay, and metabolic workflow same. Firstly, you will prepare a sample, means sample preparation, then data acquisition, then statistical analysis, right? The first thing is sample preparation is the first and most critical step in metabolomics. It ensures that metabolites are preserved and accurately represented during analysis. Objectives are prevent metabolite degradation, okay, then remove interfering compounds. See, sample preparation में what's the basics? हमें जो हमारा इंटरेस्ट है उस चीज को हम निकालते हैं. डीएनए के केस में हमने डीएनए निकाला था, और जो आरएनए प्रोटीन्स हो गए थे वो क्या हो गए थे, हमारे कंटामिनेंट्स बन गए थे. आरएनए केस में आरएनए को निकाल के बाकी सब कंटामिनेंट्स बन जाते हैं. इन दिस केस metabolites are our main focus, so we will prevent their degradation, then remove other compounds, ensures reproducibility and accuracy. So what are the key steps in sample preparation? Sample collection. Source include biofluids. You can what I would say is, you can what are the sources? Example, from blood, from urine, from saliva, you can collect metabolites, from tissues or cell cultures. Use appropriate containers and preservative to prevent contamination and degradation. Then quenching metabolic activity. Immediately halt cellular metabolism to preserve the native metabolite level. Method include freezing samples with liquid nitrogen or addition of solvents. So, what's the meaning of quenching metabolic activity? Anybody of you know what's quenching metabolic activity? पता मैं इतना बोलती हूँ, मिसप्रोनाउंसिएशन क्या कहते हैं यार, वर्ड्स मिसप्रोनाउंस शुरू हो जाते हैं मेरे. हाँ, सो कोई बताएगा मुझे क्वांचिंग मेटाबॉलिक एक्टिविटी क्या होती है? Okay, tell me the meaning of quenching word. Quenching meaning. No idea. स्टॉप करना, किसी भी प्रोसेस को स्टॉप करना. सी, मेटाबोलाइट्स कोई भी रिएक्शन होती है तो प्रोडक्ट्स बनते हैं, वो प्रोडक्ट्स फिर आगे भी दूसरे प्रोडक्ट्स फॉर्म कर सकते हैं, रिएक्ट हो सकते हैं आपस में भी. जो हमारे प्रोडक्ट बने, एक रिएक्शन है, हमारे प्रोडक्ट बन गए, वो प्रोडक्ट भी क्या पता आपस में रिएक्ट कर जाए, तो हमें वहां पे ही उस प्रोसेस को वहां पे स्टॉप करना पड़ेगा ना. उन मेटाबोलाइट्स को हमें प्रिजर्व करना पड़ेगा, राइट? उस रिएक्शन को वहीं पे हम फ्रीज क्यों करते हैं? रिएक्शन को रोकने के लिए ना. वी आर फ्रीजिंग द सैंपल्स so that the reaction can be stopped, right? Now clear what is quenching means? So we can use any chemical for this purpose, any enzyme, right? You can use cooling, heating, anything, right? Now understood? Any doubt? Any doubt? Okay, no doubt, then send me a thumbs up. Then metabolite extraction. separate metabolites from proteins, lipids, and other macromolecules. And common methods are liquid extraction you can use, like polar and non-polar phases, separate metabolites into polar and non-polar phases. Then solid phase extraction you can use. You can use a solid matrix to selectively bind and elute metabolites, right? Then sample drying and storage. Freeze drying or evaporating solvents under vacuum prevents degradation. And then you can store samples at ultra low temperature to maintain stability, right? So you have to minimize the handling to avoid contamination. These points you should these are the precautions you should take. Then ensure consistency in preparation across all samples, right? Okay, then data acquisition. Once samples are prepared, the next step involves collecting data on the metabolites using analytical technique. You can use what are the objective, detect and quantify metabolites. Then obtain detailed structure and chemical information. So what are the steps? Sample introduction. You are introducing the samples into your instruments, right? Then metabolite detection. Separate metabolites based on their chemical properties, example size, charge, polarity. You can use mass spectrometer. You can use NMR spectroscopy, right? Signal processing. Convert raw instrument data into readable formats, like in the forms of graphs, chromatograms in cases of chromatography, right? Then techniques in metabolomics, same. Mass spectrometry, measure the mass to charge ratio of metabolites, often combined with separation techniques like chromatography. You can use liquid chromatography in combination with mass spectrometry for polar metabolites. You can use GCMS. Advantage is high sensitivity and broad metabolite coverage. Then you can use NMR spectroscopy. It detects atomic, nucleic, and magnetic field to determine molecular structure. So advantages, it's a non-destructive technique and it requires minimum sample preparation. Other techniques are like Fourier transform, infrared spectroscopy to analyze chemical bonds. You can use capillary electrophoresis, separate ionic metabolites, right? Then statistical analysis. After data acquisition, statistical analysis is used to extract meaningful information from the data, right? So objective is to identify patterns and trends in metabolite level. Then correlate metabolite changes with the biological conditions. Key techniques, principal component analysis. Reduce dimensionality of data while preserving variability. See, data analysis करा, आपने सैंपल डाला इंस्ट्रूमेंट्स में, वहां से एक रिजल्ट आएगा, ठीक है? फिर उस रिजल्ट को समझने के लिए after data acquisition, statistical analysis means we have to extract the meaningful information from the data which we observed, right? So we can use principal component analysis. We can visualize sample grouping and highlight trends. Then partial least squares discriminant analysis we can use. We can commonly use for biomarker discovery. We can use visualization techniques like heat maps. Heat maps पता है मैंने क्या होते हैं? Any idea what are heat maps? जो कलरफुल वो दिखता है ना आपको, लाइक एक सीक्वेंस है, हमने एक प्रोटीन को एक कलर दे दिया, दूसरे प्रोटीन को दूसरा कलर दे दिया. अब जो पूरी सीक्वेंसेस में जो कलर शो होगा ना, वो हीट मैप होगा. So visualization tools like heat maps, volcano plots, and pathway maps help interpret data effectively. We are analyzing the data in the step. Then Metabolomics and system biology. Integration with other omics means combines metabolics data with genomics, transcriptomics, and proteomics for a holistic view of biological system. So we are combining the result now, right? Application system biology is mapping metabolic pathways, understanding gene-protein metabolite interaction. Okay, case study example like multi-omics approach to study cancer metabolism, identify synergistic relationship between metabolic and signaling pathways. Understood? Now I will ask, what do you mean by synergistic relationship? What's the meaning of synergistic? Hmm? No idea? Nobody's replying. Why? No. So this word written here, systemic biology. Systems biology meaning system biology meaning systems biology. So again, it's a multidisciplinary field, right? Interaction or combination of two or more agents, systems or processes. We have combined gene, protein, metabolite interaction here, right? हमने अलग अलग उनको combine कर दिया ना, genomics को, transcriptomics, proteomics, metabolomics, सबको combine कर देंगे तो एक system बन गया, तो हमने उसको बोल दिया systems biology. Now the word synergistic, जो interaction होती है ना दो molecule के बीच में, दो agent system के बीच में, उसको बोलते हैं. ठीक है, अब देखो क्या हो सकता है. जो रिलेशनशिप जो बना वो आगे इफेक्टिव भी हो सकता है, राइट? राइट और रॉन्ग? हेलो, आई एम ऑडिबल नाउ? सो आई वाज टॉकिंग अबाउट सिनर्जिस्टिक रिलेशनशिप, राइट? अगर दो मॉलिक्यूल्स आपस में एक दूसरे के फंक्शन को इनहेंस कर रहे हैं, मीन्स उनका जो आउटपुट आ रहा है वो ज्यादा आ रहा है एक इंडिविजुअल उससे, तो वो हो जाएगी सिनर्जिस्टिक रिलेशनशिप. ओके, देन मल्टी केस स्टडीज एग्जांपल इज मल्टी-ओमिक्स अप्रोच टू स्टडी कैंसर मेटाबॉलिज्म, आइडेंटिफाइंग सिनर्जिस्टिक रिलेशनशिप बिटवीन मेटाबॉलिक एंड सिग्नलिंग पाथवेज. देन clinical metabolomics, same role in diagnosis, like identifying metabolic biomarkers for disease like Alzheimer's, diabetes and cardiovascular disorders. Then early detection is possible and monitoring of disease progression. Then application in personalized medicine. Then therapeutic insights. So these are the two tools which we are using. These are the two bioinformatics tools for metabolomics. So Metabolomics, it's a user-friendly tool for metabolomics data analysis and visualization. Then features are data normalization, statistical analysis, pathway mapping, and biomarker analysis. And it is used in targeted and untargeted metabolic studies. Then XCMS, it's an open source platform processing and analyzing MS-based metabolic data. It detects peaks, aligns data, and identifies significant features. The application is we can identify metabolic changes in response to biological conditions or treatments. So these are the two tools. Metaboanalyst, from raw spectra to biomarkers, patterns, function, and system biology. Right, different like techniques are here. You can use LCMS, spectra, MSP. You have to select. यहाँ से आपको select करना है. Then you can input the data, you can upload the data, metadata, and same in this way. XCMS, it's an open source platform, means you can freely use this, right? Now integration of proteomics and metabolomics. You integrated different approaches. Systemic biology approach means इनका combined result आप सबका देख सकते हो, right? Key concept proteomics हमें क्या करता है? Identify and quantify protein, their interaction and modification. Metabolics के केस में क्या होता है? Metabolites को देखते हो, उनकी जो interaction है, उनकी modification है. So integration से क्या होगा? एक holistic understanding बन जाएगी आपकी. It can link genotype to phenotype by connecting protein activities to metabolite changes, right? Unveils hidden regulatory networks and you can do mapping the effect of drug treatment on both proteins and metabolites, understanding therapeutic mechanisms. Now, it is used in personalized medicine application. Then I think disease mechanism. See, I will share the slide so you guys can read in detail. The time is like very less, and I have to cover the MCQs as well. So they see same है, ये क्या है, आपको इंटीग्रेट, आपने दोनों को कंबाइन कर दिया, राइट. देन पर्सनलाइज्ड मेडिसिन एप्लीकेशन आपको पता ही है कि ड्रग डेवलपमेंट में, डिजीज रिस्क प्रोडक्शन में, ट्रीटमेंट ऑप्टिमाइजेशन में, लाइक विच थेरेपी वी कैन यूज ऑन विच इंडिविजुअल. देन डिजीज मैकेनिज्म एंड बायोमार्कर डिस्कवरी सेम है, देखो सब में ये चीजें सेम आ रही है ना. So, multi-omics approach combined data from proteomics and metabolomics to identify pathway and molecule linked to disease. It enables the discovery of biomarkers at multiple levels. Protein biomarkers are there, metabolic biomarkers are there, like indicators of disease activity. Example, C-reactive protein for inflammation, glucose level in diabetes, right? So, benefits of interaction, you can capture dynamic biological changes. Then it improves the specificity and sensitivity of biomarkers, right? Now, there are many case studies in cancer research. अगर हमें कौन सा देखना है, कौन सा प्रोटीन की अमाउंट ज्यादा हो रखी है, कौन सा प्रोटीन ज्यादा बढ़ रहा है. See, in case of breast cancer, we identified HER2 as a key therapeutic target. Then Metabolomics revealed altered lipid metabolism in aggressive tumors, right? In case of lung cancer, there is overexpression of EGFR, epidermal growth factor receptor. Then Metabolomics highlighted amino acid metabolism as the hallmark of lung cancer progression. Then colorectal cancer. Then multi-omics integration identified dysregulation of VNT signaling, proteomics, and altered short chain fatty acid production. Metabolomics. Then we can use these different proteomics and metabolomics, and then we can combine the result, right? Then future direction, we can use AI and machine learning to identify patterns, right? To make predictions. Then there will be the high throughput techniques like robotics, mass spectrometry, and automated data processing. Then there are advancement in MS and NMR. Then there are real-time multi-omics analysis. Then we can collaborate different databases, and tools are there here for the analysis purpose. Then what is the impact? Accelerated biomarker discovery, development of more precise and effective treatments. Okay, now move to the questions. You guys have to write your answers in the chat box, okay? So let's start. I think these questions are very easy. What's the primary focus of proteomics? Tell me. Write the answer in the comment, like chat box. Like 1, A, B, C, D. Okay. Obviously, proteomics means protein plus genomics. So the answer for the first question is only one person replied. What about other? You guys are sleeping? Only one person replied. Okay. B. The answer for the first question is B. So which technique is commonly used for protein separation? For protein separation, which technique is used? What's the answer for the second question? No one is responding. Why? समझ में नहीं आया क्या आप लोगों को? Okay, very nice. The answer for the second question is B. Which technique is commonly used for protein separation? 2D gel electrophoresis. We talked about 3 techniques there, right? I am from Haryana. पता नहीं पढ़ाते पढ़ाते मेरी आवाज चली जाती है. मीन्स प्रोनाउंसिएशन जो है वो मेरी गलत होनी शुरू हो जाती है. इतना बोलना पड़ता है अकेले टीचर को. इंटरेक्टिव नहीं होती ना ऑनलाइन पे क्लासेस. एफएम से चल पड़ता है मेरा ही. ओके. So answer for the second question is B. Which technique is commonly used for protein separation? हाँ जी ले लिया. Which technique is commonly used for protein separation? It's 2D gel electrophoresis. Okay. MALD TOF is a type of. It is a type of. What's the answer for the third question? Then we guys will do a quick recap in the last of 10 minutes. And what's the full form of MLD TOF? TOF means time of flight. MLD means MALDI. Nobody's writing. What happened? Okay, very good. Matrix assisted laser desorption ionization. Time of flight is a type of mass spectrometry. Right? Okay. Okay, move to the next slide, right? All clear? Okay, move to the next slide now. Question number 4th. Protein-protein interaction are studied using which technique? Protein-protein interaction can be studied using which technique? Yeah, we did this topic in detail. Ist-2 hybrid. Ist-2 hybrid technique, right? Okay. protein-protein interaction is to hybrid and co-immunoprecipitation, right? Only one person is interactive. I don't know about the others. Okay. Then metabolomics focuses on. I use very easy questions so that everyone can reply and their confidence level can boost. Okay. What about Milkari, Sarash, Khushi, Ayusha, Navinta? Why you guys are not responding? It consumes my whole day, making slides, finding questions, answers. But you guys are not focused enough. Okay. Still, only 2 responses are there. Okay. Now move to the next question. Question number 6. What does PCA stand for in metabolomics? Khushora, what happened to you? PCA stands for Principal Component Analysis. Principal Component Analysis. Okay. Next question. PCA stands for Principal Component Analysis. Now, question number 7. Question number 7. Which techniques is used in metabolomics for structural analysis? Which technique is used in metabolomics for structural analysis? Nobody is responding. Okay, Saharish, tell me about spectroscopy. And what do you mean by NMR spectroscopy? NMR stands for Nuclear Magnetic Resonance. Okay. Okay, what's the answer for that question? You guys can unmute your mics as well. You can raise your hand and I can unmute you and then you can answer the question. What's the answer for the 8th question? Clinical proteomics primarily aims at gene editing, drug discovery, biomarker identification, RNA profiling. Hmm? It's for biomarker identification, right? Answer for the 8th question is C. Answer for the 7th question is B. You can write the answer in the chat box. But I think very few people are interested in this question and answer session. Okay. Next question. Proteome discoverer as a tool for Proteome discoverer is a tool for proteomics data analysis. Okay. Spectroscopy is used in electromagnetic radiation and NMR spectroscopy uses radio waves. And radios and gas memory you have asked me, so this okay. Very good. Very good for answer. Sarish and Kush Arora both are very interactive during the whole session. Okay. So Proteome Discover, I will show you the interface of this tool as well. I have added in as a screenshot in the slides. So Proteome Discover is a tool for proteomics data analysis. And one more tool was there along with Proteome Discoverer. What was that? You guys are from which state? You mean Tamal, Kari, Kush, Sarish, Ayusha, Kushi, Sindhupriya. You guys are from which states? अब तो मैंने ये वाला क्वेश्चन ही पूछा था. Okay, Sarish is from Rajasthan. Very good. Jodhpur, Rajasthan. So you guys are doing BSc Bioinformatics. Okay. Metabolomics help in understanding what? BSc in biotechnology. So you guys are having the subject of bioinfo. Good. Biotech. Okay. Sarish and Kush both are like friends. Okay, no. Maybe senior, junior. Okay. भाई बाकी के लोग वो भी बता दो कुछ. वो कहाँ सो रहे हैं? अब तो मैंने क्वेश्चन भी नहीं पूछा था, तो मैंने पूछा था कौन से स्टेट से हो. Okay. Metabolomics help in understanding what? Protein folding, genetic mutations, biochemical changes, chromosome structure. Hmm? So answer for the 10th question is C. Biochemical changes. Okay. Now, mass spectrometry in proteomics is used for what? RNA synthesis, protein identification, gene editing, chromosome analysis. So these are used for protein identification. NMR stand for NMR stand for nuclear magnetic resonance. So you guys have to understand different techniques, right? MS, NMR, gel electrophoresis, chromatography. Okay. Now move to the next question, question number 13. Year 2 hybrid technique studies what? Gene sequencing, protein interaction, DNA binding, metabolite production. So it is used for protein interactions. Protein-protein interaction. Then, next question. The interaction of proteomics and metabolomics help in systems biology. Means you are combining and integrating more than one technique, more than one a subject. Proteomics, metabolomics, genomics, transcriptomics. So it forms the systems biology. Answer for the 14th question is B. Functional proteomics focuses on the function, right? Enzyme activity. देखो खुश. Functional proteomics means हम function की बात कर रहे हैं ना, तो जो enzymes होते हैं, proteins के बने हुए होते हैं enzymes, उनका जो function है, उनकी जो हम activity देखते हैं, वो किस में आएगी? Functional proteomics focuses on enzyme activity. Okay. Then question number 16. Which tool is used for metabolomics data analysis? हमने दो टूल की बात करी थी, right? And one of them is what's also for the 16th question? Metaboanalyst. Answer for the 16th question is A. PASDA is a method in protein sequencing, metabolomics static analysis, DNA profiling, or RNA folding. It's a metabolomics statistical analysis tool. Okay. It's a method in metabolomics statistical analysis. Answer of the 17th question is B. A key goal of metabolomics is metabolite identification. Answer for the 18th question is B. Okay, next question. Chromatography in proteomics is used for protein quantification, metabolite analysis, gene editing, RNA profiling. Yeah, for protein quantification. Okay. Answer for the last question is, answer for the 19th question is B. Oh, sorry, 19A, protein modification. Then which omics field focuses on small molecules? Small molecules. Genomics, proteomics, transcriptomics, metabolomics. Yeah, answer for the last question is D. Any doubt? Thank you for your attention. Jojo the attentive. Okay. अब इन 2 घंटों की जो अब स्लाइड्स अपलोड होगी ना, तो आपको क्या करना है, आप लोगों को पढ़ना है. जहां जहां डाउट आए, यू कैन आस्क इन द नेक्स्ट क्लास, राइट? And I think most of the things I covered in the slides. जो tools which we discussed today ना, so you have to download those softwares, then check their interfaces, क्या क्या होता है, how to upload the data in them, and how they produce the results. Okay. So tomorrow, what we are going to study is about the alignments and all. Right? So see you guys tomorrow for the new topic, sequence alignment. Tomorrow we are going to study about BLAST, PASTA and all. Right? Okay. Okay guys, bye. Take care. Okay, bye.